today's you are our first presenter of the year. Is that correct?
For 2022.
For this year,
yeah,
right?
this year.
Yes, and so I'm looking forward to hear about this imaging that actually captured my attention at Lab Days of Aarhus, and that's how I invited Sebastian. Now I'd like to introduce him to the listeners. So he studied biochemistry at Bielefeld University in Germany and pursued his academic career in Hanover at Twin Core University. Then he continued at Heidelberg University with a focus on protein complexes in viral infections. Currently, Sebastian is employed as a solutions and support scientist at LI-COR, the company which you can see in this background image. And they develop wonderful solutions. So I believe that it's a unique opportunity to hear about it from an expert like Sebastian. And with that, I give the screen to you.
Yeah. Thank you very much. Thanks for the kind introduction. I still, after 1.5 years, I'm not used to getting introduced, actually. I used to do that the whole life myself. And yeah, thanks for the kind words nonetheless. So yeah, it's really nice being here. It's actually, the talk was kind of established by the fact that we met Antonio on the lab days in Aarhus, like towards the end of the summer last year. And with we, I refer to myself and to Stephanie, who's with me in the call today here. Maybe Steffi, you can say a few words from your side first before I share the presentation.
Yeah, of course. So first of all, I want to thank Antonio that we have the chance to be here today, of course, and yeah, that we can present a little bit about, LI-COR, so our company, what we offer, our solutions, and how, of course, they might help you in your research. So Sebastian and I, we work closely together as a team. Like already introduced, Sebastian, of course, as a solution and support scientist. So from the very technical and application side of things, and I work as a technical sales consultant for LI-COR. And yeah, so this means for more, the sales part and general questions and yeah.
happy to share some insights. So I'm going to start sharing my screen, and I would hope actually that you can see the presentation right now on your screens. Is that correct? And a laser pointer? Does that match? Okay, fantastic, great. So yeah, I had quite some difficulties thinking about what I want to present today, and also about like the title that I want to give to the presentation. I will definitely try to stay within 45 minutes, and it would be really kind if you could let me know if I start going over time, because then I will shorten things. In general, as the iNano Bioscience Center is quite a diverse biocenter, I was really trying to give with this presentation the best possible overview of what you can actually do when working with fluorescent imaging. If you have any questions at any time point, you're more than welcome to interrupt. I heard we also have afterwards some time for discussing aspects. So maybe in this case, I will first go through the presentation, and then we can discuss a couple of aspects. And if anything that I'm going to show to you today is of deeper interest for you or for your research or for your group, then feel free to contact us afterwards again as well. That's no problem. So you see the title here, Exploring the Possibilities of Fluorescent Imaging with LI-COR Dyes and Solutions. I actually had a bit of help of a native speaker here choosing the title because I couldn't come up with one myself that sounded so nice. But basically what I want to talk about is what we at LI-COR can offer you as researchers at the iNano Bioscience Center and in Aarhus in general. And just to give you an idea, I do not know if every one of you knows LI-COR. So I made a small introductory presentation here about like 2 slides, just to give you an idea of what LI-COR is about. So LI-COR is a US-based biotechnology company that has been founded in 1971 in Lincoln, in Nebraska, in the United States. And our slogan since that time is impacting lives through science. In general, we have 330 employees that are distributed all over the world, with main locations in Lincoln in the US, in Bad Homburg in Germany, and in Oxford in the United Kingdom. We have two separate divisions that actually LI-COR incorporates. One is the environmental area. So this is actually the global leader for manufacturing and marketing of instrument systems for plant biology and environmental research. So if you're interested in measuring, for example, gas traces or gas analyzers, photosynthesis analyzers, this is the LI-COR environmental area. Stephanie and myself, we represent LI-COR Biotechnology, and we focus on quantitative protein assay research and small animal imaging. And we've been around by now about 25 plus years. And we develop basically everything from the reagents to the solutions, soft and hardware. And we are quite proud of the fact that our instruments are used worldwide by researchers in academia, in research institutes, governmental, as well as for us, for example, biopharmaceutical companies. Of course, you can ask yourself, okay, what basically differentiates LI-COR from other companies? Well, first of all, we were the first ones that started really looking into near-fluorescent imaging, near-infrared fluorescent imaging. So we are all aware of like fluorescent imaging, and there are some differences between fluorophores and near-infrared fluorophores, and we will talk about this. We came up with the idea of using near-infrared fluorophores for biological sciences like around a couple of decades ago. And by now we have more than 50 patents that are for imaging technologies worldwide registered, as well as more recently, we started additionally to really build on collaborations with the scientific community to build up software that allows us to analyze our assays according to current scientific best practices. We have online available a huge publication database that shows which, like in different research areas with our instruments, with our solutions. These are peer-reviewed journal articles that you can just look up online and see like what our systems and our solutions have been used for. And also we have been very active in the field of clinical trials. So we are really interested in this transition from basic into translational into clinical sciences. And we have been also very successful there. As already mentioned, and that is especially important for Stephanie and me, we really care about the support of the scientific community and of our customers. And that goes from our imaging systems to trainings to presentations like we are doing today. Or if you have more precise questions about certain applications, optimization processes, we are available at any time points to be contacted. Those are our contact details. I'm also more than happy to provide them after the presentation again to Antonio. I don't know if you maybe can share them somewhere or if that would be of help for us, absolutely no problem. We will arrange that. And what we will talk about today, and I will go through, is a couple of different aspects. I would like to start talking about the detection chemistry. You already heard near-infrared imaging is what we are specialized on. From there, I would like to talk about our dyes and solutions that we can offer, show some applications and some assays, just to give you an idea of what you can do. But you yourself know much better how maybe our solutions and dyes might be useful to you. Last aspect, if I manage to get there, would be data integrity. So that is what we are recently working on, really focusing on data integrity, software development. How can you support the scientific community in generating reliable, reproducible data all around the globe? I would like to start actually from our point of view that we ask ourselves the questions, when you're doing experiments, what do you as a researcher want from your experiments? And as a matter of fact, I do believe, having worked in science myself for a long time, we want to be confident in the data that we are generating. Because if you're confident about the data, you're confident about presenting your data and publishing your data. More importantly is also that if you generate data, you want to show that they have a significance. On the one hand, of course, a significance for your research topic, but on the other hand, also like that you are able to reproduce your results. And this is exactly actually not only what you want from your data, but this is also what reviewers and journals want from your data, because that is what we have to think about being and working in the scientific environment. If you're just keeping your data for yourself, it's not about that. It's about publishing, it's about communication, networking, discussing results. And all these things that I've been mentioning so far are exactly the same principle for reviewers and journals. And of course, especially when we talk about like reproducible and significant results, we have to have a look on protein assays, and we have to ask ourselves, how can we actually generate reliable and reproducible results in biomedical, biological sciences? And there are a couple of aspects that we have to talk about. And it all starts with the fact that every one of you who ever worked in a quantitative fashion for sure wants to have a robust normalization strategy, because you're not only looking at your protein of interest, you're also looking at a loading control, at an internal loading control you can use amongst a couple of different samples to normalize your target protein too. Something that's also very important when it comes to reproducibility is the signals that you're working with. You can work with chemiluminescence, bioluminescence, you can work with fluorescence, near-infrared fluorophores. Important is you have to have accurate linear quantifiable signals in the assays that you are working with. And lastly, of course, that is something that goes beyond the whole community, is you want to generate reproducible, reliable results. And those are also reproducible, not only from experiment to experiment, but they should be reproducible from another user somewhere entirely else that tries to recapitulate what you've been doing. As a matter of fact, not only we at LI-COR think that reproducibility and quality standards are absolutely essential in biological sciences, also a couple of scientific journals and publishers have set quality standards for protein assays in the past years. And just to give you an overview, this is a list of things that are important to the Journal of Biological Chemistry, but also the Nature Publishing Group, for example, has very similar standards as the ones written here. So if you go through that list and have a look on it, we actually realize, yes, normalization strategies, technical biological replicates, you know, these are things every one of us is aware about. But there are also some other things that are really essential. For example, antibody specificity and selectivity, demonstrating linear range of the detection method. You know, those are things, maybe we have heard about that, but the question is if we all stick to these rules at any time point, or if we are even aware of these aspects. And this is what also the next few slides are going to be a bit about. And that's when I want to start with the detection chemistry. And I would like to start actually very basic with something every one of you has heard at one stage in their career, and that is chemiluminescent imaging or chemiluminescent detection, short ECL, is like the most basic, the most like traditional method, maybe with isotope labeling together, how you can actually generate protein assay data. The principle is you have your assay in which you have your target protein or antigen, and this is recognized then by a combination of different primary and secondary antibodies. For chemiluminescent detection, these antibodies are linked to an enzyme, the so-called horseradish peroxidase, HRP, and signal generation is done by addition of a substrate, and the substrate is of course converted by the enzyme into a product and emits photons of light. And those photons of light are what you have been measuring in the past with a film, with a digital system, with a camera system. But what is so important when we look back to what we actually want from our research is the question, when we work quantitatively, if there is a direct proportionality between the signal response here and the amount of protein that we have used in our assay. And as a matter of fact, for chemiluminescent detection, it's not that easy because we work with an enzymatic reaction. And I'm absolutely convinced every one of you knows that enzymatic reactions are depending on a couple of different variables over time. So timing, yes, is one aspect, Another one is the temperature. The temperature has a massive impact on the enzymes that we're working with for chemiluminescent detection. And if we, for example, stay with Western blot protocol, just for example, we also have to deal with different substrates that are available, and that means we talk about different kinetics. At the end of the day, that basically means that the signal proportionality to the amount of antigen is not a necessarily given aspect for these reactions. And there is another aspect that limits chemiluminescent use, and that is the fact that we only have one channel available. The light that is generated emits at around 400 to 500 nanometers of wavelength, and there is no second chemiluminescent channel. So we can only look at one protein at a time in the assays that we are working with. If we now compare that with fluorescent detection, the principle stays the same, just our secondary antibody changes, and it changes to a fluorophore instead of an enzyme that is now linked to our secondary antibody. And these fluorophores, they can be excited by an external laser source and then emit photons of light. And again, the so important question, if you want to work with a quantitative assay between the linearity of the signal here and the amount of antigen that you have on your assay. And as we are working with a static fluorophore, and also in our assay, the amount of antigen is static as we have maybe fixed the cells or fixed the samples using a defined amount of DNA or RNA, we do not expect any variables. So a fluorophore is independent from the time point in which you look at the reaction. It's independent from the temperature. If it's like 18 degrees room temperature or 31 degrees, it doesn't really matter for the fluorophore. On top, that then means that we have this direct proportionality. And another important factor, especially for protein assays nowadays, is if we have different sources that can excite our fluorophores, we can use several fluorophores at the same time if they do not crosstalk to each other. And that is referred to as multiplexing capacity. And this really can also enhance your time because you have to perform less assays than you previously thought. One quick look at the timing of those two reactions before we continue. I mentioned the chemiluminescent detection is kind of variable. And this is very well visible if you actually start using two individual proteins, one being highly abundant, one being low abundant. If you compare the ratio in a chemiluminescent reaction of an assay that you perform of these two proteins to each other, you will actually realize that the ratio differs depending on the time point which you take to look at this reaction. In comparison with fluorescent detection, you have the advantage that at any time point, the ratio between these two proteins is the same. And of course, talking about reproducibility and reliability, this is the better way to image your protein or DNA, RNA assay data. Something that comes along very often from our point of view is actually that people are asking us, hey, can you be sometimes, for example, for membrane-based assays, can you be as sensitive with fluorescent imaging as you can be with ECL, like chemiluminescent imaging? And as a matter of fact, yes, you can be as sensitive as chemiluminescent imaging. The difference is just we're talking about fluorophores. So we have to take care that the background that we have has to be low to achieve a high fluorescent sensitivity. For example, in any form or way of experiment where we use fluorophores in a microscopy setting, it's exactly the same. As long as you can keep the background level low, you have a good sensitivity. For imaging with protein assays or DNA, RNA assays, it's exactly the same. So how can we achieve that? There we go now more into the direction of what LI-COR is doing, because I mentioned we are working with near-infrared fluorophores. When I talk about near-infrared fluorophores, I mean fluorophores that have their excitation emission spectra beyond 650 nanometers of wavelengths usually. And that differs massively from visible fluorophores, for example, like EGFP, the SiDye family, or big parts of the Alexa fluor family. And why is it so important which fluorophores you actually use? Well, the thing we have to talk about, the topic we have to talk about is autofluorescence. So everything that we are working with in a laboratory setting, let it be tissues, cells, samples that are lysed, the buffers we are working with, the chemicals, the plastic where we are working with, everything has an autofluorescent capacity. And the autofluorescent capacity can be differently strong at different wavelengths of light. And as a matter of fact, for many things, if we work in vitro, working with near-infrared fluorophores, there is nearly no autofluorescent capacity and a low autofluorescent background at 700 and 800 nanometers wavelength. This is an example from a microscope, but we can also go very practically into assays that we do in the laboratory. For example, membrane-based assays or plate-based assays. What you can see here is the relative autofluorescent capacity of different types of membranes, as you can use them, for example, for protein arrays, Western blot experiments, or DNA or RNA-based assays as well. And what you see here is the further we go towards the near-infrared wavelength of light, we have 100-fold lower amounts in the autofluorescent capacity of those membrane types, for example. And that for you means that you have to overcome less of a hurdle in this case to be sensitive with the assay that you're working with. And if you keep those things in mind, then we can actually go back and talk about multiplexing of experiments in vitro. And I just wanted to give you an overview of what you can actually do with LI-COR fluorophores and LI-COR dyes and solutions here. So what you see is a fluorescent-based multiplexing approach, either, for example, on a membrane assay, on a plate-based assay, or if you want to work ex vivo, for example, with tissue slides or microscopy slides. This is how it can then look like, and this is the impact that it has when you work with near-infrared fluorophores. Something that is also very important in this case is that this principle with the low background fluorescence not only works just in vitro, it also works very well in vivo. So with the near-infrared wavelength of light, you can see here and have a look at this graph. So what you see here is the extinction coefficient of hemoglobin. So a very prominent protein that we will find in animals, as well as like some extinction coefficients of water, for example, here. And what you see here is the near-infrared imaging window for in vivo imaging. And what you can see actually is that there is a very low autofluorescent background also for in vivo imaging. And just to give you an idea of how strong this impact can be, here's a comparison between a Psi 5.5 dye and our IRDye 800CW. So what you can see here is the mice in both cases have not been treated with any compound. So here you have the fluorophore diluted in an Eppendorf tube in both cases. And this is the autofluorescent capacity at around 650, 700 nanometers of wavelength of a mouse that have been just fed with the regular diet, usually chlorophyll or those kind of things like also beta carotene. They have a high autofluorescent capacity. And this is what you see here in the intestines of this mouse. If we go towards the 800 nanometer channel, again, you have a low background fluorescence, and that makes it much easier to use near-infrared fluorophores, really not only for basic research, but also for translational or clinical research. This brings me to the second part where I want to talk about the dyes and solutions that we are offering. There is a broad variety of assays and dyes we can offer, and I try to go through most of them, but even that are not all of which we have actually available. I would like to start looking at near-infrared fluorophores, which is our IRDye family. And what you will see here is just a small overview in which channel of, for example, an Odyssey imaging system, so of our imaging systems, where can you image these fluorophores. And then here you have the fluorophore name and the excitation maximum and emission maximum wavelength in nanometers. So what we commonly work with at LI-COR is the 800 nanometer channel. I already mentioned that. And you see we have different fluorophores available here with different excitation and emission spectra around 800 nanometers of wavelength. And why we have those different ones available, I will explain in a minute. But then the second big channel that we are using is our 700 nanometer channel, and those are fluorophores that excite and emit around 680 to 700 nanometers of wavelengths. And again, you see here different fluorophores listed. In all cases, the advantage is, as mentioned, we have a low autofluorescent background, and that goes from basic in vitro research to ex vivo and in vivo research as well. What can you now do with all these different near-infrared fluorophores that we have available? Well, we have a variety of protein labeling kits as well as click chemistry kits. So what it basically does, or what you can do with this, is that you can label your, oh, sorry. Do you see yourself right now? Because you here, okay. Yeah. Sorry for that. Okay, so let's continue. Sorry. So we have different labeling kits available to label, for example, antibody, proteins, or peptides, as well as DNA and RNA. But we also then, for example, have the dyes available in a form that you can link them to larger macromolecules. And this is exactly what you will see here on the next slide. So here's now the same list of dyes available that I showed you previously. And here what you can see is, so first of all, we have different dyes available that are linkable with in form of an NHS ester, malamides, or carboxylates. So those are usually considered to be working with, to be linked to proteins, peptides, or antibodies, for example. We also have these dyes available, as mentioned, for different forms of click chemistry. So if this is a topic that interests you, you can actually link our fluorophores via DVCO, azides, or alkenes to your biomacromolecules if you want that. And then lastly, as mentioned, we also have our fluorophores available as phosphoramidites. And phosphoramidites are very effectively binding to DNA or RNA strands towards the 5' end. And this can be of use, for example, if you want to label specific DNA or RNA fragments. In this case, let me already tell you now, if you're interested in DNA or RNA work with near-infrared fluorophores, we collaborate with IDT, so Integrated DNA Technologies. It's a company from, I think, Germany or somewhere in Europe. I don't want to mention it wrongly. And they allow directly linking our fluorophores to custom-made DNAs and RNA fragments. Another thing, and that goes again towards more like the in vivo levels, we also make use of our IRDyes in case of imaging agents. So we have different imaging agents available that allow you in vitro as well as in vivo to look, for example, into tumor imaging, into inflammation or arthritis, as well as some more specified research topics like structural imaging or cell trafficking. And some of these applications I also will pick up later on again in the applications area when we talk about that. What I would like to do now, and that goes more into like your direction in general, is, as I do not know everyone's research topic, I wanted to shortly talk about optical probe development. In general, optical probe development refers to the fact that you yourself have a target in mind that you would like to label and use, for example, for, I don't know, microscopy work. You can use it for cell-based work, for in vivo work, but you haven't figured out how you can visualize your target for your research. And this is something that LI-COR has invested quite a lot of time in, and we have a variety of technical and application notes available that have recommended workflows if you want to label your protein of interest or your molecule of interest. And you will find a lot of information in these technical papers that will help to provide some best practices for optical probe development, and especially also will help you save time, money, and frustration, because we already went through that at LI-COR. In general, if you are interested in working with a macromolecule or with a target that you have in mind, there are 3 steps in the development of how you can visualize your target protein and using LI-COR fluorophores. So the first thing is, of course, you have to have an idea of what you actually want to target. It might be a compound like a peptide, an antibody, a drug, but it might also be, for example, DNA or RNA. This is all possible. And what you want to do is you want to link your probe with, if you want to go into translational research, a clinical suitable dye. if you just want to stay with basic research, it's also okay to take a non-clinically suitable dye. And then this is what you have to do first. And for that, of course, the IRDyes are a fantastic fluorophore to use, because as mentioned already, with the high signal to noise ratio, low autofluorescent background, and this very versatile use from in vitro to in vivo, you have a lot of advantages using these fluorescent dyes. Plus, we might be able to support you a bit if it goes towards clinical trials. We have some experience in that, so we can also give some advice and support in this direction. After designing your probe for your specific research project, of course, you have to validate that your probe is working well. And that usually in biological science is, of course, done via in vitro assays. So, for example, you would start assessing the specificity or the binding affinity of your labeled probe in vitro. So, for example, in a cell-based assay, and there you can, for example, use one of our imaging systems like the Odyssey DLX or the Odyssey M. Also, if you want to go more into a clinical direction, you, of course, next have to assess the specificity of your probe, the biodistribution, clearance, etc. And for that, we have actually a small animal imager, which is called the Pearl Trilogy. Next thing would be to also, for example, assess ex vivo. You want to make, for example, some tissue sections or some microscopy slides. And also for that, we offer solutions. And I would like to show some of these solutions. So cell-based validation assays is something that is commonly used by many researchers. We at LI-COR have defined the terms in-cell Western and on-cell Western assays. So it's basically in a multi-well plate, you want to look at your probe, and you can, for example, determine the binding affinity or its specificity. Those response experiments can be performed with this probe. And for that, we also have our two imaging systems, the Odyssey DLX as well as the Odyssey M. And just to give you an idea, what are these imaging systems? For all systems that we at LI-COR are working with, the Odyssey family are laser-based scanning systems. So we not only provide you with the fluorophores, but we want to make sure that very specifically only the fluorophores are excited if you use them in your assays. And for that purpose, you need a laser-based system, because only laser-based illumination allows a very narrow excitation of just the fluorophore and not of the surrounding or the probe that you've been using. For that, we have these two instruments. The Odyssey DLX actually allows accurate detection and quantitation of cell-based assays. It's a pure near-infrared imaging system, so 700 and 800 nanometer laser that allows to really fastly in a high throughput fashion allows for generation of high quality data. It fits up to 6 microwell plates. And then we have the newest addition to our family, which is the Odyssey M imaging system. This one actually only fits 4 microwell plates, but it has a line scanning technology. So again, it works with lasers, and in total it has 18 channels, of which 4 are fluorescent based. And it allows much faster scanning time. So you have the advantage that you can even have a higher throughput, for example, for your in vitro probe validation. And then just to mention for the in vivo part, there would be the Pearl Trilogy, which is like an animal imager. And this is also specifically to be used with near-infrared fluorophores. However, it could also, for example, look into GFP expression or BLI expression. Another thing is, of course, microscopy slides and tissue section imaging. I don't know how many of you are working in general with microscopes. I see a hand that goes up there. So something at least that I found very convenient from, if I look back at my time, sometimes you want to see in which of your samples a specific protein is expressed, and you're sitting like half the day in front of a microscope. And this is something that with the Odyssey M definitely belongs to the past, because this imaging system has a market-leading 5 micron resolution. It's not a microscope, it's an imaging system, and it has a true one-time magnification. What this allows you to do is you can put your samples into this microscopy slide holder up to 12 at a time. You can put it in the imaging system and you just let it scan through. You can triage through your slides. So the imaging system captures your images. You just look through the images afterwards and you decide which of those samples are worth going to the microscope. And this can really take up a lot of time off your plate and really allows to focus on something else in the meantime. Last step of the optical probe development, of course, would be if you want to do your research fashion in a broader scale, then you have to scale up the manufacturing. For this, we can still help you. We can provide some support. If you then go into clinical trials, we can unfortunately not provide the right dyes anymore, so you would need to partner with someone else. But at least until like this point of time, we can fully support you in the steps that you are doing. This was very narrowed down from what we can actually do. I mean, you can see we have a lot of technical papers available there. And yeah, if you're interested, I will provide a follow-up email, I guess, to Sadek and to Antonio, so you have access to this afterwards. Or if you're further interested in that topic, you can, of course, also contact us as well. One just quick view on the imaging systems. I mentioned them on the side. So our imaging systems are the Odyssey imaging systems. As you can see, we talked about the laser-based scanning ones. That's the Odyssey DLX and the Odyssey M. We also have a camera-based system that also works with lasers. So at any time point, if you want to have a laser-based system, our Odyssey family always works with lasers. And just one thing that I specifically want to mention here, and we are actually very proud of this part, all of these imaging systems have a dynamic range of 6 logs. And what that basically means is when we talk about dynamic range, we talk about what is an imaging system, detection hardware, able to capture in a single image acquisition, not with like different exposure settings, in one single image. And those 3 imaging systems, they capture 6 logs of data. That basically translate into more than 22-bit floating point TIFF files. And if you want to have it precisely, it's more than 4.2 million grayscales that every image is captured on these systems. Independently, if you work with like tissue slide, with a Western blot, with a plate-based assay, it's always 4.2 million grayscales. And that is absolutely market leading for any imager of any imaging family. I will skip a bit through the more detailed descriptions of the 2 imaging systems because I would like to show you some applications as well in the 45 minutes that we have. We talked about what the systems can do. Just in general, be aware, for example, for the DLX, we have more than 15 supported applications, and you will see some of them coming up soon. It has a very broad imaging surface, 25 times 25 centimeters, where you can put many tissue slides, multi-titer plates, or Western blots, whatever you can think about DNA, RNA gels. Choose whatever you want. The Odyssey M is entirely new, has on top to the fluorescent laser channels, AP and trans illumination channels, so it can also capture gray, like black, white, gray scales, as well as like colored samples. And it has an entirely new imaging detector, which is called the scientific CMOS detector. And it really fasten up your research because, for example, it's 50% faster than the Odyssey DLX. If you think about a regular plate, microscopy imaging, a regular 96 well plate, it will be able to scan a regular 96 well plate at, I think it's 10 micrometers resolution in less than 10 minutes. So you don't have to sit next to it. It's fantastic. And yeah, we will come to the applications in a minute. In general, keep in mind what I mentioned, 6 logs, 4.2 million grayscales in every image independently, if it's a membrane, a plate, or a tissue slide. And if you work with biological relevant samples, 6 logs actually also translate into you don't have to worry about saturation. especially for membrane-based assays, we have never observed any saturation in biological relevant samples, because 4.2 million grayscales is more than any membrane translated into keeping proteins would ever be able to do. One last aspect to the imagers before we come to the applications is actually that all imaging systems we are working with, and also our software, does not introduce any form of image manipulation. We know that there are imaging systems out there from much bigger companies that, for example, apply binning processes, flat fielding processes, or stacking processes, and those things are not performed with a LI-COR imager. Every time you capture an image on one of these imaging systems, the image will be raw data exclusively. So at any time point, you also work with your raw data, and this is what we can promise from our point of view. Okay, so after talking about the detection chemistry and talking about the dyes and solutions and the imaging systems, let's talk about and give you some ideas of what you can actually do with all these dyes and all these imaging systems. And just to give you a fast, quick overview, what can you, for example, do with our Odyssey imaging systems? You can perform membrane assays like Western blots, visible, colorimetric, chemiluminescent if you want, also possible. But you can also, for example, do GFP assays, M-cherry assays. You can look at multi-well plates. We talked about this, the so-called in-cell or on-cell Western assays. Some of the systems can measure ELISAs, some form of cell health assays, GFP reporter assays. We can look into protein gels, nucleic acid gels, reporter gene assays, and we talked a bit about slides as well. And that's what I just want to show you on the next few slides. Just to give you a broad overview, here you can see just near-infrared Western blot experiments, or like those where you combine the different fluorophores from visible and near-infrared channels. But also you can, for example, use fluorescent protein gels, and here you always have the channel selection on top. So if you are imaging Comassi protein gels, if you, for example, go for Cypro gels as well, those can all be imaged on our imaging systems. Colorimetric protein gels like silver stains, Comassis, can also be imaged in other channels like the trans illumination channels, for example. And something that I wanted to shed a light on for today's meeting was also nucleic acid gels and nucleic acid applications. So not only can you image your nucleic acid gels on some of the imaging systems, but more importantly, something you can do, I already mentioned, we collaborate with integrated DNA technologies to build custom-made DNAs and RNAs with our different IRDye fluorophores. And maybe some of you have heard about so-called EMSA assays, so electromobility shift assays. Basically, what people might be interested in is the interaction of RNA or DNA fragments with individual proteins, and you use this in a gel-based setup. So basically, like DNA or proteins migrate through a regular SDS page with a certain speed because of their size. If you combine them together, it's a bigger complex migrates slower through a gel, and this can be perfectly visualized when, for example, using DNA fragments or RNA fragments with our IRDye fluorophores. And just to give you an idea, for example, here, researchers were interested in binding of a wild type DNA sequence toward or a mutant DNA sequence. So here is the wild type sequence, here is the mutant sequence. You can see those are the controls where we just use the DNA or the DNA fragment with the fluorophore. And then here are different bacterial strains, as far as I remember. And you can see if they bind either the wild type or the mutant version of this DNA fragment. And this is, for example, how you can employ our IRDye fluorophores with DNA or RNA assays. But this is just one example of many that you can perform. On top, we also talked about plate-based assays that you can do with these imaging systems. You have here two-colored or three-colored applications. ELISAs can be imaged, though we have to be fair and have to say that ELISAs in our case can be imaged on the Odyssey M imaging system. However, they need to be analyzed outside of our software packages. Applications like fluorescent tissue samples or microscopy slides I also already showed you. So this is just again a pickup of what I already had said. And then also, for example, stained microscopy slides or H&E stainings, Alcyon blue, these can all be imaged. And again, here we come to this like triaging. So you can put them all at the same time, up to 12 slides on the imaging system, and the imaging system does the work for you while you just do something else. Towards the end, one thing I wanted to show you, and this now goes really into advanced settings of what we can do with our IRDye fluorophores. I mentioned also in vivo work is possible. And what you can see here are different imaging agents like the 800CW EGF optical probe, the RGD optical probe, or the 2DG optical probe. Those are probes for tumor imaging, either, for example, for tumors that overexpress EGF receptors, that overexpress integrin receptors, or that show elevated sites of glycolysis. And I also know from some customers that use, for example, these dyes not in vivo, but rather in their like advanced 3D culture settings, for example, this can also be done. So basically, in this case, it is up to you what you do with these dyes and with what kind of system you're working. And last application I want to show you is that we also have what is called the IRDye bone tag. So this is a tetracycline derivative that is a calcium chelating compound. And this is very well incorporated into mineralizing bone. And you can see here how this then looks if you, for example, have an animal. You can very nicely see here that this dye is incorporated into mineralizing bones. So yeah, there are a lot of different possibilities that you have. As we are reaching the 45 minutes, and I definitely would like to also give you the chance to talk to us and to discuss with us some potential interests. Just very shortly, besides everything that we have been talking about, the solutions and dyes we offer and the applications, I mentioned that we collaborate with the scientific community to set standards in regard to software development. And this is basically, since a few months, we have some new softwares, and that is based on the fact that data integrity for protein assay analysis is a big issue. We have variation in the same data that we provide different researchers with. And that's why we started developing a software that is based on these guidelines and quality standards I earlier mentioned. And yeah, this is the Imperia Studio software. It can be used with LI-COR imaging systems. And we really developed this software to adhere to the current scientific best practices. We talked to the Journal of Biological Chemistry. We collaborated with Nature Publishing Group. And the outcome was a workflow-based analysis tool for any form of protein assay that you can image on our imaging systems. I will not have the time to go through the software, but I'm more than happy to answer questions to the software if there are any. And we basically did that to really automate and streamline to get reproducible results in between different researchers all over the world using our imaging systems. Advantage is that you now use one software package for all the validations and the analysis of your protein assay that you're looking at. And with that, I'm actually at the end and wanted to thank all of you for your attention. We are more than happy, independently if you have our imaging systems or not. We are more than happy to support you in your research, in the research area you're working. We can provide you with different protocols and white papers that are available from our point of view for different applications, imaging system, fluorophores. We have every now and then really exciting webinars that go into different topics. And yeah, we also personally, Stephanie and myself, are available for you to discuss any of the topics we talked about today. And with that, I'm actually at the end of my presentation, and I hope it was helpful and interesting for you and for your research topic. Thanks.